So our next speaker is an abstract selected speaker. It's a graduate student, Scott Hickey from the Hammond Lab at UC Berkeley, and he's going to talk about the development of high-throughput screens to target SAM1 riboswitches.
Okay, great. Hello, good afternoon. I'm Scott Hickey. I work in Ming Hammond's lab at the Department of Chemistry at UC Berkeley. And so just in case anyone's wondering, the Cal Bears did not make the NCAA basketball tournament this year. So the only March Madness that I'm experiencing is just being a fifth-year grad student in the lab. So I'm going to tell you a story today about our development of high-throughput screening assays for targeting riboswitch RNAs. And so riboswitch RNAs are actually structured RNAs that control gene expression in bacteria. And so they do this by binding to and sensing small molecule metabolite ligands. And this binding event actually results in a conformational change of the RNA, which leads to differential gene expression of a downstream gene. And so this is the switch part of a riboswitch because it can switch between two states depending on cellular metabolite concentrations. And so in the normal function, or under normal conditions in the cell, the gene, a downstream gene, for example, would only be repressed when metabolite concentrations are very high. So the cell is not going to waste time and energy making metabolite that is highly abundant. But in contrast, so now if you hit these riboswitches with a drug molecule, you end up repressing this gene, downstream gene expression, regardless of metabolite concentration. And so for this reason, riboswitches are a pretty convincing target for antibiotic development. And also, the added bonus is that they're only found in bacteria. They're almost exclusively found in bacteria. So they're a great target for antibiotic development. But again, if you hit this riboswitch sequence with a drug, you turn off the ability of the cell to respond to metabolite concentrations, and this would lead to cell death. And so thanks to the very large amounts of genomic sequencing data we now have, it's pretty well understood that riboswitches are a widely utilized regulation strategy, genetic regulation strategy in bacteria. And so this is just a representation of the phylogenetic map of a certain class of riboswitches known as SAM1. And we know that riboswitches control greater than 2% of all genes in Bacillus subtilis, and they've also been identified in many human pathogens. And so we can think about developing broad spectrum antibiotics that target a riboswitch class in all bacteria, but it's perhaps more or also equally interesting to think about selectively targeting riboswitches only in pathogenic organisms. And so can we take a riboswitch that's found in a pathogen such as Staphylococcus aureus and another riboswitch sequence from a commensal bacteria such as Lactobacillus, and can we develop screening assays and methods to screen for compounds that are selectively target pathogenic sequences over the non-pathogenic organisms? And so this is a pretty exciting idea, but it requires a lot of challenging work in that you have to end up screening a large compound library of small molecules and also potentially screening a large RNA sequence library as well. So you're kind of screening a library against another library, and this is very difficult to do. And so we're kind of looking at methods of high-throughput screening development to enable this type of experiment. And so there is some precedence for compounds that can selectively target riboswitch sequences in specific organisms. And so the FMN binding riboswitches is a family of riboswitches that bind to this compound, which is flavin mononucleotide. And there's a known natural product, Roseoflavin, that actually can differentially target FMN riboswitches from different bacteria. And so one of these sequences in a certain bacteria binds to Roseoflavin or a metabolite derived from Roseoflavin, and then another sequence does not bind Roseoflavin. And so we want to know if we can exploit this potential of the sequence diversity between riboswitches in the same family by developing compounds that target specific riboswitch sequences. So high-throughput screening approaches take advantage of large and diverse sets of compound libraries, and there has been some very nice work done targeting riboswitches using high-throughput screening approaches. So for example, a molecular beacon assay was developed for targeting adenine riboswitches, which is based on transcriptional read-through of this riboswitch. And this has been used to screen adenine analogs for the adenine riboswitch. And also an equilibrium dialysis method has been employed to screen the TPP riboswitch, screening a fragment library against this riboswitch sequence. But there's a lot of work to be done in this area, particularly because this idea of screening a library of compounds against a library of RNAs. So for example, this molecular beacon assay is great for targeting or for screening compound libraries, but you have to develop a molecular beacon for each particular riboswitch sequence that you want to screen. So it's not good for screening diverse sets of RNA sequences. And on the opposite, on kind of the other hand of this, the equilibrium dialysis assay can screen diverse sets of RNA molecules, but it requires the use of only certain types of compounds for this assay. So we're kind of interested in developing new technologies for high-throughput screening approaches targeting riboswitches, and I'm going to tell you about two of them today. One of them is using fluorescence polarization in a competitive ligand displacement assay, and then the second is actually a collaboration that we are working on in the development of a microfluidic mobility shift assay that we're pretty excited about. So our model system for targeting these RNA riboswitches is the SAM1 riboswitch family. So the SAM1 riboswitch is a set of riboswitches that bind this molecule, which is S-adenosine methionine. And this is a very important cellular cofactor. And so the other reason, one of the reasons why we're choosing to target this riboswitch is because it hasn't previously been targeted for therapeutic development. But we use structure-guided design so our idea was to target this riboswitch by using a fluorescently tagged SAM analog as a ligand so that we could perform fluorescence-based assays. And so we used the crystal structure of this riboswitch, and particularly taking a look in the SAM binding pocket, we were able to design a compound, a chemical analog of SAM, where this top part is the SAM mimic, and then it's linked at the 3' hydroxyl to a fluorescent molecule. And so we actually, I won't have time to talk about the synthesis at the moment, but I have a poster tonight, so if anybody wants to talk synthetic chemistry, feel free to stop by. And we're also using this synthetic scheme for SAM analogs for different types of SAM analogs, in particular as methyltransferase inhibitors. But we're very happy to see that this compound actually is able to target SAM1 sequences from different bacteria. And so we took basically 3 riboswitch sequences from different bacteria, including one from the pathogen Staph aureus. And we are looking at, here I'm showing just fluorescence polarization binding curves in vitro. And what we see is there's an increase in polarization of the probe as it's bound to RNA. So as we increase RNA concentration, we see an increase in the fluorescence polarization signal. And we've also used this assay, or this compound, to develop a competitive ligand displacement assay. So now we're actually starting with a high fluorescence polarization signal, and then if we add in a competitor that's known to target this riboswitch, in this case the SH sulfone, which is just a SAM analog, we see a switch from high FP state down to low FP state, which represents the ligand being displaced from the binding pocket. And so this was done in low throughput format at first, but we've recently done this also, translated this into a high throughput format, 384-well high throughput format. And so now we're just looking at 16-well replicates of our control data. And so without competitor, with no compound added except for our fluorescent compound, we see high fluorescence polarization. And then if we add in SAM or an analog of SAM, which is known to target this riboswitch, we see a decrease in the signal representing displacement of the probe. And we use adenosine, which has very, very weak affinity for the riboswitch, as a negative control. And so we've also actually taken this screen now and done a very small screen, a preliminary pilot assay with a 320 compound library to target this RNA. And we're actually following up on this preliminary hit that we see. But we quickly realized that this assay requires a lot of RNA material. And so this is okay for screening hundreds of compounds, but if we want to screen thousands of compounds against multiple RNA sequences, we need to do better. So we think that this FP assay actually will be well suited for a secondary validation screen, but we need to do a little bit better for a primary screening system. So we're turning to a different type of assay for the primary screen, and this is actually a mobility shift assay where it's taking advantage of the fact that riboswitch RNAs actually compact upon ligand binding. And so traditionally you can separate a bound and unbound riboswitch by native gel electrophoresis, but these assays typically require long times. And so we actually developed a microfluidic platform in collaboration with Amy Herr's lab in the bioengineering department. And this is a pretty impressive assay because we can run now these mobility shifts in 12 seconds per run, and it only requires attomolar amounts of RNA per lane. And so this is a pretty nice assay for doing these types of screens. It's very quick. And we've actually taken it forward and done just a preliminary low throughput screen of different SAM1 riboswitches from different bacteria. And so we took basically 5 computationally predicted SAM1 sequences from various organisms, and we used our assay to demonstrate that we could see a mobility shift for all of them. So that validates the fact that they are actually SAM1 riboswitches. And in terms of developing a high throughput format of this screen, our collaborators in the Herr lab have actually already developed a 96-well format for this assay where they can run protein separations in less than 10 minutes. And the preliminary data that we have for looking at RNA separations looks promising, and it only uses about femtomolar concentrations of RNA, so we're pretty excited about that. And so in conclusion, I've told you about 2 assays for targeting riboswitch RNAs, and we think that the microfluidics platform is actually pretty promising as a primary screen for screening compound libraries against libraries of RNA sequences. And then we also have this fluorescence polarization-based screen that we can target SAM riboswitches as a secondary validation assay. So any compounds that we target, or any compounds that we identify using these screens, we would then follow up using in vivo validation methods. So I'd just like to thank the entire Hammond lab, especially my PI Ming for her wisdom in the chemistry realm and also in the RNA biology realm. And I just wanted to point out that Tanya, who's actually here, has a poster tonight, and she is working on an entirely different project in looking at plant immune responses. So just all of these sources for funding and everyone for their attention. Thank you very much.
Any questions? So maybe I missed it, the microfluidic system, is that in-house developed, or where does that come from?
Yeah, so that's in a collaboration with Amy Herr's lab, who's in the bioengineering department at UC Berkeley. So it's just a collaboration with that department. But yeah, they do everything in-house.
Okay, well, thank you. Oh, sorry, one last question here.
So the 300 chemical compounds that you use, they are some similar structurally, or you just want it?
Let me repeat the question.
Yeah, so the question is the 320 compound library structurally related to the metabolite? Yeah, so the metabolite is SAM, and it's S-adenosylmethionine. And the compound library that we chose to just do an initial screen was actually a kinase-targeted library that is ATP heterocycles. So it's adenosine mimics in a way. So it is sort of targeted for adenosine-based recognition. Yeah.
Okay, well, thank you once again.